não, porque você vai querer que as pessoas sentam aqui na mesa? Não, né? Porque vai ser a apresentação. Não, agora vai apresentar. Eu vou querer um copo. Para, traz um copo, por favor. Let's start. I think we can start. We are online already. So, good afternoon. It's very nice to have you here, the visitors and the people from the lab. So we start now the BRAVO seminars. We'll have 4 very, very nice international presentations. And we start with the presentation of Professor Dora Ventura. Professor Dora Ventura works in the Department of Experimental Psychology at the University of São Paulo. You all know her. I don't think I need to tell too many things about her, but she's been working with basic and clinical research in the area of visual neuroscience. She got several awards in Brazil and internationally. And it's my pleasure to introduce her. And she will be talking about mechanism, genetics, and visual performance, clinical research in humans, and animal experimentation. Thank you very much.
Thank you. Thank you, Mirella. Thank you all for being here. Welcome, our international guests, Álvaro, Aurélie, begin with an A, Cyril, and Jan. It's all in alphabetical order. Thank you very much for having come to our country, and I hope you're enjoying now and will enjoy throughout your stay being in Brazil. And what I thought of doing today was the idea was to present our lab, but instead I thought it was better to present our thematic project at FAPESP, since you are members of that project. And I don't even know if you received a copy, but if so, it was in Portuguese. And I doubt that you had the patience to go through it. Maybe Álvaro. But so I'll give a bird's eye view of the project. And what I'd like to tell you is that FAPESP has 3 distinct lines of projects. There's a regular project in which one researcher presents an idea and then a course of action to explore that idea. And so that's the regular project. And then there's the second level, that's the thematic project, which is a group of people present an idea or several ideas that are intertwined. And so that involves several people. It varies depending on the topic and around the topic. And then there's CEPID. It's a Center for Research and Innovation. The regular project, I think, is a 2-year grant, or maybe 3. The thematic project is a 5-year grant, and it's larger. Every year you send in a report, and at that time you can ask for more equipment or whatever you need. And then there's the CEPID, that's a 10-year project, which involves even more people, well, the thematic project may involve several universities, as you're going to see at the end of my presentation. And so we are in the middle. Here, as you have in projects in the United States, Europe also, there's a PI for the project. So that's my case. And there's a co-PI, which is Marcelo Fernandes da Costa, faculty member of our lab, of our department. And then there's what I decided to call, it's not in FAPESP's organization, but our project leaders. These are people that have been working in the project and have been, well, to distinguish them from the other people, they have been supervising at least one student. And these are Daniela Bonsi. Daniela is going to be here a little late. She is at a meeting. And Mirella Barboni, who just spoke to you. Einat Hausmann. André Lieber. Where's André? André. And Leonardo Henriques. So these are people very involved with the project. We have in our vision animal and human electrophysiology, human psychophysics, neuropsychology also. And we have connected with the animal electrophysiology, retinal morphology, and molecular genetics. So we started the neuropsychology when we're working with mercury intoxicated patients, and they had very bad symptoms, and apparently to decide that the person is intoxicated, the medical decision is made through a neuropsychological assessment. So that's why we started investing a little bit in neuropsychology, and now we have been doing that for a number of reasons. And when we look at vision in humans, we look at visual acuity, temporal resolution, color, contrast, movement, dark adaptation, pupillometry, ERG, and visual evoked potentials. And we have special protocols that have been developed by Ian on chromatic, on-off, and to study Magno and Parvo pathways. And in animal experimentation, we look at opsin genetics. So this was something we began later. We felt the need for genetics, and we sent Daniela to the United States to the Knight's lab. She spent a year there. And then when she came back, FAPESP gave us a genetics lab, which now is run by her and by Einat. We studied the topography of retinal cells, evolution analysis methods, photoreceptor immunohistochemistry, and retinal histochemistry, and other morphology techniques, like even Golgi staining we have done successfully. It was very nice. ICEF protocol of ERG and other protocols for ERG is chromatic and on-off. And so this is the animal experimentation. We're now beginning with a little bit of behavioral experimentation. In the past, we had a lot on honeybee behavior. The lab is over 50 years old, and we should write history of the lab because it has been through so many things. But this is how it is now. The experimental models we use are human and animal. And the human animals, pandemic viral infection, neurologic and psychiatric diseases, development, so old age and young age, aging, aging is there too, genetic diseases, and we have, of course, the control in healthy subjects. And the animals, you can see them there, the mouse, the lizards, snakes, and a little bit, we're beginning with owls now. And Jan will have that experience. I wish I could be there too. Scientific questions that we have in human research, we study mainly visual function. And we are trying to ask what is the impact of disease, aging, exposure to neurotoxicants, and other insults in the visual system in order to find more about how the visual system works. And not only that, but in order to be able to tell if the disease is progressing or to diagnose it. How can we optimize testing in the lab and the clinic is another question that we ask, because a lot of information we have is done in the lab, and to transfer that to the clinic is a big task, and it's not possible because the just this. Yes. So, and the other question we ask is about biomarkers. What indicators can best reveal the impact of disease or insult over the visual system? And what indicators would allow early diagnosis and follow-up? So, in animal research, we ask what are the genetic characteristics of color vision in different species, and how do they contribute to understand the evolution of color vision? And also, how is the retina organized in different species? We have been studying that quite a bit using snakes as a model. And does its topographic organization reflect its environment or habits is a question. In the beginning of this line of research, which was brought to us by Einat as her interest in her previous work with snakes, I thought it would be very difficult to demonstrate. I really did not believe we would be very successful. But I was very wrong, and we're able to show in the retina plasticity that's due to habit or to the place the animal lives in. And another interest in the animal research is if performance, meaning electrical responses or other index of performance would reflect improvement with treatments, because biomarkers can be used to diagnose or to follow up conditions that we have. So a summary of the project is that it aims to investigate the visual system through sensory and perceptual tests with psychophysical methods, visual electrophysiological protocols, color and contrast vision are being investigated, visual perimetry, dark adaptation, pupillometry, in basic research and clinical applications for early diagnosis and follow-up of ophthalmological, neurological, and psychiatric diseases. In pupillometry, you are mainly concerned with assessing functions of the melanopsin pigment. And color vision and genetic determination of visual pigments in different species are being directed at investigating evolution of color vision in vertebrates, with emphasis in what I was just saying, environmental pressures and circadian habits. And animal visual electrophysiology is being used to test and evaluate toxicity and efficacy of new medical drugs injected directly into the eye, and functional recovery after treatment with genetic therapy for Duchenne muscular dystrophy. So we have been doing that. We include that as a project, but it's being done in France by Cyril, mostly Andrés involved, Mirella, Jan, and Álvaro, directly with the project, and Aurélie, and that is within one of our projects, a project in foreign collaboration. We're also developing new psychophysical methods to various purposes in the understanding functional of cortical areas, attention, and movement perception. Also, we study the vision of old patients and of patients exposed to pandemic viral infections such as Zika and COVID-19. And we aim to develop new tests and to apply existing tests in different populations. Because if we're creating new tests, we have to test them and validate them and so on, previously not examined. So looking at all the sub-projects we have, we developed, we organized them in 4 axes. One is the development of new visual tests to introduce adaptations that allow use in the field in clinical situation, reducing time of testing and equipment cost. And to monitor the functioning of cellular types, these are the electrophysiological tests, neural pathways and projection areas, employing available morphology and physiology knowledge in the design of these tests. Another axis is visual evaluation of patients and controls to validate the tests that will be developed using comparison with gold standard tests, to detect disease diagnostic biomarkers, to obtain early evidence for alteration of functions, and for follow-up. In animal experiments, that's also one of the axes. Invasive, we have animal experiments because there we can perform invasive experimentation. The animal models we have are natural through mutation or induced by genetic or other procedures. And we use ERG to evaluate the integrity of different cell types in the retina. What I previously said, medical drug toxicity and efficacy, and function recovery after drug or genetic therapy. And here we distinguish the very promising results in Duchenne muscular dystrophy. The last axis is molecular genetics, to identify color vision genes in patients to establish if losses were congenital or acquired. We begin with that because in studying color vision, we could not state in some cases whether the losses were congenital or acquired. And we found that there was nowhere in São Paulo, or even with Luis Carlos, with whom we collaborated quite a lot in Pará, there was no lab that could perform these screening tests for us. So this is why Daniela went to the Knights' lab and learned the genetic procedures, and we now can do it in our own lab. And of course, this part complements and explains the results of visual psychophysics of color vision in humans. The first experience we had with that was when we published, submitted a paper on toxicity of in gas stations of what you call vapor from gasoline fuel, and in our gas stations, the cars are how do you say, are fueled by people, not by the owner of the car, but there's one employee that does that all day, and so he exposed to vapors constantly, all day long. And we found that there was a high loss of color vision in that case. And we submitted the paper, and it came back with the question whether two of our tested subjects were congenital, or was that an acquired loss. And lo and behold, it was acquired. We could prove it because we had the genetics lab beginning at that time. So we were very happy. That was the first time we could use the lab to confirm that this extreme loss that was seen and thought that could be congenital was not, was acquired. So also we do basic studies about color vision in different animal species and their evolution up to human color vision. And we evaluate, so the molecular genetics is involved in that, and evaluate the adaptive capacity of the visual system to environment and species habits, because understanding this plasticity is relevant for understanding evolution. So these are the projects we have. We have 3 groups, group 1, 2, and 3, and we have in each group several projects in which that's how we organized. This is visual evolution of patients. So the first project involves COVID-19. The second project involves 3 sub-projects of here. 3 sub-projects here of psychiatric diseases, neurodegenerative diseases, and this has not started. Neuromodulation, we haven't begun, but it's one project that we are interested in beginning, in which pupillar light reflex and visual electrophysiology will be studied. And so this is mostly in macular degeneration, and Luis Baran, who's here, has a postdoc to perform these studies. Dark adaptation in DMD patients, where's Sarah? Yes, so she is working this project, and also here. The optic neuromyelitis syndrome disease and positive anti-aquaporin 4G immunoglobulin serology is a project that was proposed by Mirella. It's being done at Unifesp Medical School of another university in São Paulo with Ana Laura Moura. And the storch, Z. civilis, toxoplasmosis, rubella, cytomegalovirus, and herpes simplex is the doctoral work of Leonardo Aparecido, who's not here now, is finishing that. And this is a study of vision in babies, newborn children, babies and children. Reading and numerosity is being studied by Marcelo. It's probably in his presentation. And developing new tests. This was very exciting, was visual function automated ocular tracking based on its software written by Diego Lima, the student, and he based his doctorate in that. A low price solution for eye tracking. And we also have color and contrast vision in a tablet so that we can take it to a clinic and to a hospital, and instead, rather than bringing the patient here. This visual acuity and contrast sensitivity with multiple stimuli. This is a test which involves a complex stimulation, and Marcelo will explain it in his presentation. Also the same with white and pink noise, which these are stimuli that are superimposed in gratings, and so they become complex stimulation, and so the results are compared to plain gratings, and these development of psychophysical tests and for humans and non-human primates is also Leonardo. And here these are some projects that Marcelo will be talking about. And the animal experiments, we have mouse models of Duchenne, mouse model of Usher syndrome, mouse model of diabetic retinopathy, rabbit model for toxicity of drugs, and ERG in reptiles, lizards, and snakes. And also we've begun ERG in fishes, zebrafish and tilapia. And this is in retinal electrophysiology, and this structural and molecular basis of vertebrate vision, retinal morphology, and genes of the visual processing pathways. So retinal circuitry of snakes, this work with the lizards, differences in male and female, and they have different habitats also, but they meet, I guess. Opsin gene evolution in reptiles. This project was not begun. It was going to be done with research in the Amazon, but unfortunately we lost him to COVID. This was very tragic. Evolution of visual pigment genes in mammals. This is Daniela doing, and melanopsin in owls. So the methods, we have ERG, we have different protocols, and VP also different protocols, and we can study children using this portable equipment, which is very handy. And we have behavioral methods, visual acuity, using teller acuity cards, and this new test developed by Diego Lima of ocular tracking. Color vision, we have 3 tests, Cambridge, the CAD test from Barber from England, and this new test that we have, tablet color vision test. And there's some new tests that are being developed by Leonardo in which the subject will track the saturation decreases along a path, and the subject should go with the finger and track the path. Very interesting test. And also contrast sensitivity. And this is the dark adaptation equipment that Sarah knows very well. And this is how we measure chromatic pupillometry is when we study melanopsin using a red light and comparing it with a blue light, in which the return to the dark adapted size is much slower. So we measure that return, and that would be an indication of active melanopsin. And animal methods, we have ERG, in which we have several protocols, and these are examples of snake ERGs. And we have methods of morphology and molecular genetics for the identification of cellular types through immunohistochemistry or staining. And we also have a very patient counting and to see the topographic distribution. And here is short wavelength, a long wavelength, and merge to see cells in which you have both together, or the set of all receptors. And we have molecular genetics, different molecular genetics techniques. And here the counting and topographic distribution. And the expected results are in all areas: human evaluation, new tests, animal genetics experimentation, animal models, would be the association of genotype, phenotype, and Duchenne muscular dystrophy, visual losses as biomarkers in psychiatric and neurologic diseases, infectious congenital diseases in newborns, school difficulties in dyslexia and dyscalculia, are most of what the things are that we would be doing. And new tests, the visual tracking test that I mentioned to you, new color vision tests, and second order perception and attention mechanisms that Marcelo will explain. In animal and genetic experimentation, evaluation of efficacy, the main is the evaluation of efficacy of genetic therapy for Duchenne muscular dystrophy, and animal models and their contribution for the knowledge of color vision evolution and its plastic relationship with the environment and species habits. And with all that list of projects that I showed you, we have forming people where we have 13 PhDs underway, 6 master's degrees, and soon we will not have master's degrees anymore. It will be eliminated from our, I don't know if I like the idea, but that's the plan of the university. And we have 4 undergraduates working with us. And these students, they are 19 students, they have 17 of them have fellowships, which allow them to stay here most of the time. And 3 of the 4 undergraduates also have fellowships. Now, our national and international collaborations. And we have a lot of, this is in Brazil. So Federal University of Pará, both departments here, of Rio Grande do Norte and of Paraíba, of Minas, and several other institutes at USP, Unifesp, which is another medical school, and the medical school in Jundiaí, where we did a lot of work in the hospital, because they have a maternity hospital and pediatric, and we did a long-term study of Zika virus infected children there. And internationally, Canada, Panama, Hungary, France, Germany, United Kingdom, and Sweden are collaborators. So Germany and France are here. Hungary is here too. And Einat was the person who had a very prestigious fellowship, Marie Curie Fellowship, and went to the United Kingdom and to Germany. And Marcelo has a link with people in Sweden, and that's it. So here we thank FAPESP a lot. This is Marcelo, and the people that are project here leaders are all smiling at us here. And I thank you very much and tell you that we like to party a lot. Marcelo's.
Thank you very much. I think we can leave the discussions and questions for the end. Yes, besides being this very active professor, researcher, scientist, she's a great mentor as well. So after my 20 years together working, I think I can say that it's everyday learning with her. Thank you so much. So unfortunately, Marcelo couldn't come. We'll have a video from him. I hope it will work. Let's try to start.
And they were supposed to trees. Because is using differentiate from the sensory measurements and also using the measurements made for other modalities, such as for phonoaudiology, they have the central pathway, central auditory processing. And of course, this helps in clinical communication between professionals if you use similar terms. And the objective of central visual processing measurement is to evaluate low and higher order perceptual phenomena. As well, we can also do some measurements for spatial visual functions, as you can see here. The idea in the central visual processing is to functionally seek to trace the path of construction of our visual percepts, and also relating the area or brain areas that are strongly suggested by the literature that are related to their processing. So we can be able to identify this battery the level and the magnitude of impairment if occurs. So it's a nice kind of assessment for neurological psychiatry patients. The central visual processing that we develop here in our labs composed with both static and dynamic visual functions that enables to investigate a wide variety of functions in both dorsal and ventral streams. And the dorsal stream here is presented in light pink, and the vector streams presented in light green. Some examples of perceptual functions include radial acuity. It's one radial acuity, perception, visual texture, figure ground by gestalt closure, shape distortion, and also we have coherent motion and spatial memory for position, for example. And some examples of spatial function we have here: the numeric estimation, also automatic attention, sustained attention, voluntary sustained attention, and visual memory for digits, and velocity of reading speed. The main groups of patients that we have been testing here include autism spectrum disorder, in which literature show a strong reducing phase procession, reducing in inhibitory mechanisms. So some of them have huge response to sensory stimulus. They also have reduced motion perception, and also reducing in quantitative and abstract visual processing. For the attention deficit and hyperactivity / impulsive disorder, we have been testing the two main core of functions, one related more to automated attention, in which the literature showing us reduced peripheral and spatial attention, and in some cases this population could also have a quantitative processing reduced. That's a comorbidity about 40%. this both condition. And for the deficit, the children with attentional deficit, in fact, the protocol of sustained attention had been shown that the literature shows that for these children that we tested sustained attention protocol, they have reduced feature detection and also reduced visual search for spatial stimuli. For learning disability, we have been working with two main groups, dyslexia and dyscalculia. Dyslexia is related to problems with reading acquisition, in fact. And they can learn, they can understand, but reading they can get the main concept of the test. And we also have literature showing that these children have a reduced visual span of the parafoveal information. So this one would be one of the main problems in children with dyslexia. And for dyscalculia, of course, the problem is number sense, spatial relationships, as those we found in geometry, and also in quantitative manipulation. And more recently, we have been working with Down syndrome children, and the recent literature showed that they have been any kind of problem, including motion perception, spatial location, form organization, emotional face processing, also numerical processing problems, reading problems. So you can find any kind of problem in Down syndrome, and the level of the problem is much more correlated with the phenotype expression of the genetic disease. So just to finish my presentation, summarizing what we have been found, some of this information is under publication and other coming up. For visual processing disorders, we have been found reduced function for statics visual functions like contrast sensitive luminance for linear, radial, and texture stimuli, and also for form organization, distortion, and discrimination, subjective center defined by line bisection, but all the more dynamic functions is normal in this kind of children. Visual processing disorder sometimes mimics learning disabilities, and we have been found a group of these patients during our evaluation. For the ADHD children, the hyperactive/impulsive ones, we have been found problems reducing performance in linear luminance, texture luminance, form organization discrimination, form discrimination, coherent motion, and visual span for reading. But we have found higher than normal thresholds and sensitivities to temporal contrast sensitivity, automatic attention performance, and also for time perception. They have shortened time perception. They are accelerated children. And for the children with the classical attentional deficits, we have found problems in more sustained or more statical functions, including line section, form, discrimination, organization, distortion, in our protocol of sustained attention, visual memory, coherent motion, and some of them also they have a lower reading velocity. In dyslexia, we have some problems with form, but line section, coherent motion, and are the main found in the perceptual problems. They also have much more problem for the reading function, and similar in dyscalculia that we have problems, some problems here for perceptual organization, perceptual level for as form organization, coherent motion, but you have a lot of problem numeric estimation, number line, and time perception. For autism children, they have generally an increase in contrast sensitivity function, mainly for the lower and lower meter spatial frequency. That's that corroborates what the literature says about the hypersensitivity they have for some sensory mechanisms. We have been found this increase in contrast sensitivity luminance, but a reduction in contrast texture. So it means that between sensory and perceptual lower level, we have a bottleneck effect, and it has become a biomarker for the autistic children. Also, we have problem with form organization, line deception, coherent motion, numeric estimation, time perception, and here we can also test the higher level of higher functional level of these children. So down autistic syndrome, children with level 1, 1, 2, we can test this function, and for higher levels, which means worse functional abilities, we sometimes could only test this introductional sensory or even lower level perception function. And for Down syndrome, we can basically found anything alterated. We not have so much children with Down syndrome tested, but it looks like that they have different kind of problems, and these problems could not be also related with the other abilities. Since we have children that could read, but they have very strong reduction in this kind of lower level measurements, and we have children that could not read, but they are very nice in this perceptual function. So we cannot say so much now about the Down syndrome children, and I guess that's all. Thank you for listening during these 15 minutes. Sorry again to not be present today, but of course, see you in the lab during this week. Bye-bye.
Okay, thank you. Do you agree with a coffee break, or it's better? Okay, then for the people watching us online, we will be silent here for 15 minutes, and we'll be back soon. Thank you. With Álvaro, Professor Álvaro Rendon. I will put it also. Sara, vê aí. Alô, alô, alô. I think it's just this microphone. Alô, alô. Esse aqui não. Alô, 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 alô. Dois. Não, não. Só para deixar. So both microphones are working, both of them. And you prefer that thing or this one to go to the next slide? Which one you prefer? To change the slide.
I think I prefer with this
Okay, then I turn.
because I have my fingers are so large that I always missing. Okay?
You know what I will do? Just a minute. I take one chair from here because then.
Yes, it's better. But yeah,
And you have more
Can-
space here.
okay. And then you use like the okay,
Just to test.
okay.
Okay. Good afternoon again. So to the audience here and the online ones, I would like to introduce now Professor Álvaro Rendon. Professor Álvaro Rendon is an emeritus professor and one of the founding members of the Institut de la Vision in Paris. His pioneering contributions were instrumental in establishing this globally recognized center dedicated to advancing research on vision and related neurological mechanisms. He also works closely with the Neuroscience Institute in Paris-Saclay, where he focus on, his focus is on the study of dystrophin DP71 as a key protein in brain and retina development and cognitive functions, particularly in the context of Duchenne muscular dystrophy. Professor Rendon's research has significantly advanced our understanding of how different dystrophin proteins influence the development and function of the central nervous system in animal models. His work utilizes sophisticated molecular and gene therapy approaches, including AAV-based strategies to restore DP71 expression. This groundbreaking research opens new therapeutic avenues for addressing cognitive deficits associated with DMD. As an expert in molecular neurobiology, Professor Rendon continues to contribute to the global scientific community, providing deep insights into the intersection of neurodevelopmental disorders and therapeutic innovation. He's also coordinating educational programs to strengthen the student exchanges between Mexico and Europe. In 2014, Professor Rendon was here, visited our lab. He was presenting at a BRAVO symposium, so 10 years later again. That was in Caxambu at that time, and we had a lot of meetings, and he was fostering collaborative exchanges that enriched both teams' research perspectives. Professor Rendon's extensive contributions to molecular neurobiology is a bridge between basic science and the therapy for the patients, making him a key figure advancing treatments for neurodevelopmental disorders. And today's presentation will be about tracing the evolution, the history, and development of DP71 in the visual system. Thank you very much, Professor.
Hola a todos voces. Estoy muy feliz por estar en Sao Paulo, esta tercera edición de la DECO dis Retina. And now, please allow me to speak in English, because even if you can understand my Spanish, okay? I think that English is our better language. And first of all, I would like, of course, to thanks Dora, that she has been one of my. We were collaborating in somehow also friendly. Do you know? 20 years ago, when we went to Arvo, in the Arvo we were 3 people talking about dystrophy, Duchenne muscular dystrophy in retina. It was Dean Pillers, it was Dora Fix Ventura, and Alvaro Rendon. And we were a little club since that time. And now I am really very happy. As you can see, I am not a younger man, a younger researcher. And also, I would like to thank Mirella, because thanks to Mirella, we make a true bridge between your lab and my lab. We have publications together. We have been, okay, it's a kind of friendship that between France, Brazil, and also a little bit of Mexico. And I really thank you very much for this invitation. And I am very impressed by your introduction to the lab, your first talk, because it's so modern that really, and I think that your plans, I admire you to have this projection in the future, okay? And I see you will succeed bringing all these young people, okay, to follow your example, okay? Thank you, Dora. Thank you really very much. And now, as she told me, I want, since, do you know, in France, there is very special, the emeritus, they have very special appointment. They don't need to have, they don't allow to be lead a team, don't have students, don't work with money, nothing at all. But I did all that thanks with all this collaboration in the world, that I have the opportunity to develop, okay? And of course, Brazil, one of them. As Mirella said, 10 years ago, I was here already, okay, for a nice meeting. And now here is not a title, okay? You can see that it's not a title, okay? Is, okay? But what I will show you is, okay? I will show you tracing the evolution, the history and development of DP71 in visual system. Okay, we work from the very beginning with all the dystrophies and so, but little by little, you will see how DP71 becomes a start in the retina. And that's what I will do now. But for the story, I think a lot of you don't know the story of dystrophins and who discovered the dystrophin, the Duchenne muscular dystrophy, and also all the story till arrive till now, okay? And that's what I will try to give you. I hope that I won't be too fast. It's 4:25, okay? I am started 25 minutes later. I think that I won't stop at 5, okay? Because in principle, I had 1 hour, and I hope that, okay, I will go faster, okay? But now, since here's the history, I bring the history to now who discovered the Duchenne muscular dystrophy. Okay, this was a man that is Guillaume de Boulogne, okay? Duchenne, that becomes Duchenne de Boulogne, and that the first paper, okay, in 1886 was called, and when he described the so-called now Duchenne muscular dystrophy, he, okay, do you have a pointer? Okay, anyhow, okay. No, don't be worried. Okay, that's right. That's cool. No. Okay. Okay. Where should I push for the light?
You can just
Okay.
the middle one to see the laser.
Okay. Okay. Ah, yes. Okay. Okay. In 1868, he made the initial description of Duchenne by Duchenne. It was the initial description by Duchenne of the muscular, and this he already reported that this pathology have muscular and cognitive deficit, but he didn't mention any visual impairment. And we have to wait almost more than 1 century to the group of Louis Kunkel in United States. They discovered the gene responsible for this disease, and they called them the Duchenne muscular dystrophy. Okay, and in 1992, okay. What happened?
Well, I found it here. The one I used.
Ah, okay.
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 So here is.
Yes, but do you know do you see my fingers? They are too large, and they are always pushing a lot, okay? And then I will try to don't mistake. That's the way, okay? Okay. Okay. That's what Mirella proposed me here. But look at that, okay? In 1992, the Michael Koenig, that was a postdoc, Alsatian postdoc, because I did all my beginnings in Strasbourg. I was living in Strasbourg, France for more than 30 years, and that was there when I started to work with Duchenne. And in 1992, Michael Koenig in the Kunkel laboratory, he made the complete sequence of the dystrophies. And I predicted that it was a cytoskeletal proteins. And since I was a cytoskeletal that time, that I was a specialist in cytoskeleton in brain, I used to move little by little to Duchenne. And okay, in 1992, and that's important point as well, a 71 kilodalton protein that is a major product of the DMD gene was discovered and reported by the labs of Jaffe and Nudel, okay, the labs in the Weizmann Institute. And now, let me just to give you a small rappel and a small recall. What is, I think a lot of you know that, okay, but you know that this is is located the gene in the region XP21, and but that Duchenne is the largest gene that has been discovered till now, more than 2 million pair of bases. And the important point is that it has at least, I said at least, 7 promoters that will produce, that were synthesizing 7 different proteins, 2 that are called DP and the molecular mass, okay? And they are so-called DP427, DP260, DP140, DP116, and DP71. They have an N-terminal, a central domain, and also a cysteine-rich domain and a C-terminal, I told you. And today, we will talk only about our star, that is DP71. I won't describe anything about it with the others. I just do something that I always saying, and that Cyril, he said that I am too, how can I say, too tough for for ask that is that the people that are working with the gene, they say the isoforms DP260 is a isoforms of the gene. They are not isoforms. They are products of the gene because they have their own promoter. Okay, and this should be clear. Okay, they are not isoforms. If you get courses or you write papers, don't say isoforms. Even our the big friends of Duchenne that are in England, they say isoforms. Okay, but it's wrong. Okay, now we go into this story. Okay, this is I show you here a historical perspective of dystrophins in nervous system. We didn't. I will try to use this. Okay. Where is, yes, okay. In 1993, in my laboratory, Daniel Jung, PhD students, he showed for the first time a Western blot published in 1993 of the DP71. And then we not only stay working with the brain. At the very beginning, we were exploring who other models that could express dystrophins. And also with collaboration with a Mexican group in 1996, we discovered the expression of dystrophins and the DP71 mainly in PC12 cells. PC12 cells are model cells of neurons, and that's well utilized as a model cell. Okay, and also when in collaboration with Algeria, this is in collaboration with Mexico, in collaboration with Algeria, now she's professor in Alger, and Latifa Dorbani, we discovered dystrophins in neuroprophysial lobe of normal and dehydrated rats. You should, I don't know if you follow I think that you follow the literature, and that's very impressive because after it's the only lab in the world that are working with dystrophins in this neuroprophysial lobe, and they discovered that it has a role in the astrocytes that are moving during the dehydration, and then the DP71 is playing a role, and that's something that we didn't expect at that time. And now let me show you, okay, two papers that are also in the nervous system, but that had been done. That's in retina, because the ampullars, as I told you, our colleague, our friend of Dora and myself, in 1993, we published the first paper of the dystrophin expression in the human retina is required for normal function. And now she's still on the way. And with Mirella and Jan, we met her this year in Argo. Mirella was so impressed that he was sitting beside Jan, and she was just, she couldn't speak, okay? That was. And also, I think that you will have the evolution of this. You know that somebody called Cyril Vaillant, he published the first paper in the literature of the influence of dystrophin gene mutations in the mice MDX in the behavior of these mice. And that was a very important paper, because till now, 30 years after, is still quoted. Okay, and then of course you will hear about now or what he's doing now. Okay, and now let's go to the and also in 2007, okay, Marcelo that you used to hear about, okay, that he and with Dora, they show the for the first time the impairment of the red-green color vision in the MD. That was very important, and I was very impressed. And there I met, of course, he's not around now, Marcelo, but I wanted him to go to do his postdoc with me, but he preferred to stay here and continue some ophthalmology work. Okay, now let's go to this part, the historical perspective of DP71 in division system. Okay, for the first time, the first paper that published a differential distribution of dystrophin in the retina were Thomas Claude-Pierre in my laboratory. Okay, here you see that the P71 is very important already. He showed that. And then in the same student in the 2000, okay, we show for the first time that DP71 in the retina was located in the Muller, specifically in the Muller glial cells. But we still looking for where the dystrophins, and the first paper that published that the Duchenne and DP71 and dystrophins were found in the zebrafish come from my lab, from a Mexican postdoc, Francisco Bolaños, and he showed that DP71, it's the development of retina in the zebra in the retina was different. It depends more of DP71 than the dystrophins. And with this, I think that you have now an idea that historically we can be very 30 years ago for this work, and now we're still being here. Okay, now let's go a little bit further. At that moment, okay, and now that we knew very well the DP71, here I used to show you that DP71, okay, and is the most expressed dystrophin products that are in the mouse, okay, in the mouse and in the man, okay, in the mouse retina and in the man, okay, here, sorry, okay, and from this, okay, before going further, I told you that, okay, they are products, okay, but the Mexican laboratory, in the Mexican team that are working between Cyril and myself as well, we found they discovered that there are at least all these isoforms of the DP71, and the main difference that they found that was the presence of the exon 78, and they call this group of dystrophins with other mutations, with other differences, isoforms. They call the group D, and this is the group F, and this is a small group different. And you can see that in the brain and the retina, the isoforms D are more richer in the brain than in retina. That means that both proteins have a different functions, okay? And I think that with working with Cyril and with the Mexicans, we realize that, okay? And now they are following even the role of some of these isoforms of DP71 in the development of models. So first in the PC12 cells, and now in brain as well. And they have papers, and that's really they open a new field with the DP71 because they don't have the same function, okay? These isoforms, and we are really, I think that for Cyril and for me, we are really very happy because we still collaborated with them. Okay, now I go back to the story, okay? And the story, historically, we have the chance when we decided to work either with the function of DP71 in a mouse model, that the group of urine noodle and oh, sorry, did you see? That's my finger. How? What can I go back? Okay, this, yeah, okay. And David Jaffe and Uri Nodel, they with a graduate student was Rahel Sarik, they took 3 years to produce this mouse. These mice were the first strain of DP7 of dystrophins that didn't express DP71, but all the other products of the DND gene still functional, okay, still working. And that was very important for us. And they replaced a part of the DP71 first exon by a beta gene, okay? And Cyril and me, we were very lucky because they were non-neuroscientists. They believed that DP71 was very important in the development of brain. And they found that there was not a difference between the cow DP71 and the normal, the wild type. But since Cyril and me were so interested in that, that they allow us to work myself with the retina, with the vision system, and Cyril with the brain. And that was an important step in the development of our research. Okay, and now I am really starting with the story of DP71, and the first question that we ask is the localization in the mouse retina. And what is the experiments that we did? We compare with a pan-specific antibody against all the dystrophins, the expression in the retina. You can see that they are in the outer plexiform layer around blood vessels, in the inner nuclear layer also around blood vessels, and the inner limiter membrane, okay? And in the here's something that looks like astrocytes, okay? And when we did the same experiment, the same in the retina of DP71 null mice, you can see that the labeling disappeared all over, and just we had the labeling of all the other dystrophins at the level of the outer plexiform layers, and that all that disappeared, meaning that we deduce from this that the Mueller glial cells and the astrocyte, this DP71, really now we have the evidence that DP71 was located in the Mueller glial cells and the astrocytes. And now, before going to show, before that I show you what is the function of DP71 in the retina. Let me in the Mueller glial cells. Sorry, just give you a little recall of what is the molecular function of dystrophin DP427 in the muscle. Okay, it's an anchor protein. That's what you call a anchor protein that will link all this complex, okay, of disoglycans, of sarcoglycans, okay, and calvulina, sarcospan, and between the inside the cell and outside the cell, okay, and the laminina, the alpha dystroglycan, it's anchored in the laminin, and this is the function, or this is the function of the dystrophins in the muscle system. And what we show when we ask ourselves what's the molecular function of DP71, okay, to have it, do you know, in a retina we couldn't have a purified part of the Mueller glial cells, okay, because they will be mixed with everything. In that case, Patrice Ford and Abdullah Sen, they cut the retina here, okay, here, and we use only the end feet, the glial cell end feet and the astrocytes of here to show that the complex that of the DP71 in the Mueller glial cells. And you can see here that we found two kinds of complex. One, okay, that is dependent of DP71 and is linked in F-actin. This is the complex that will fix the agrin, the laminin, okay, by the beta dystroglycan and the alpha dystroglycan. but also it links, okay, no, okay, that anchors, okay, in a part of the plasma membranes that is rich in cholesterol. It anchors K4.1 and AQP4 that are very important channels for the function of the dystrophins of the retina. Sorry. Okay, and we also show that in the Müller glial cells there is another complex that it has. The anchor protein is this analogous of the dystrophins that you know, of course, that is the utrophin, but this is the big utrophin because we have also UP71. In this case, is the big utrophin that is able to anchor only H4.1, but AQP4 cannot bind to this part of the complex, and the rest of the complex is similar to what we found for P71. Okay, and that give us the idea what could be the function of this complex in the in the retina, and how we try to what the question that we ask now, okay, is what are the phenotypes of the DP71 null mice in the retina. And for the first answer that we give to that is we study the dystrophins of the distribution of K4.1 and AQP4, okay, in retina. Here you can see in the wild type and the DP71 null mice, okay, this is we're labeling with bimantine the Müller glial cells, and here in the wild type K4.1 you can see that it's in the merged image. If we can maybe, I don't know if you can, we can take up this, just, okay, it's better, but don't sleep, please, okay? Because now you can see here that really it's the K4.1 colocalized exactly, okay, in the Müller glial cells, okay, and as well as AQP4, okay, here, okay, and what happens in the DP71 null mice? In the DP71 null mice, you see that K4.1 is not anymore located around the blood vessels, and it's more diffuse in all the retina, okay? And what happens with AQP4? It is not only diffuse in the retina, but also its expression has been inhibited. Here I show you, okay, this Western blot in which we see then the wild type, the comparison of the expression of K4.1 that is not changed in the for K4.1, but the expression there is not anymore expression or very small for AQP4. Okay, that means that what we observed with the complex is it was true. That means that K4.1 and utrophin is still there, and AQP4, since AQP4 cannot bind to utrophin, it's inhibited its expression in the DP71 null mice. And this is one of the first phenotypes that we found due to the absence of DP71. But there is also a second one. You know, here we compare the morphology of the Müller glial cells in the wild type and the DP71 null mice. Okay, and here it's the DP71. You can see, okay, that it's labeling, and in the end feet it's okay. It's well contracted like that, like a small ball like that. And what happens in the DP71 null mice? Look at the end feet. The end feet is looser and is spread out. That means that the absence of DP71 even modify the morphology of the Müller glial cells. And of course, this is also one image, but we did a statistical analysis to show that really that is true. The morphology of the DP71 of the Mueller cells in the DP71 null mice is completely different. You can see there, and that will modify a lot the relations with the rest of this of the cell, okay, of the compounds. And this is one of the modifications that we observe. As you know, the Mueller glial cells have the end feet are fixed, they interact with the blood vessels. Sorry. No, that's, yes, okay. Sorry, okay. And this is the same image, okay, in the DP71 null mice. Do you see that this morphology certainly affects the relation with the blood vessels, and they are much flat, the interaction between the blood vessels and the Mueller glial cells. That means that this contact, it's completely modified. And what happens with the astrocytes? Here I found you a more what we did with to study the morphology of the relations between the Mueller cells and the astrocytes. Here you see, I don't know if you there is not anymore like this one, okay, in the middle. Yeah. Oh yes, okay. You can see the interaction of the end feet of the astrocytes with the vessels, okay, that is completely modified when with the astrocytes of DDP71 null mice. And there is not only the number of there is also here we estimated the number of astrocytes in the number of astrocytes in a retina, and they are less astrocytes in absence of DP71. Okay, and as you can see, the network of vessels, it's completely modified. They are more spread and they are more larger. And we estimated here the number of vessels, okay, the density of vessels, and you can see that it really diminished in absence of DP71. As far as the development concerns, this is at P6, the development of the vessels in a flat-mounted retina, and you can see that at 6 days, there is really a delay in the development of the vessels in absence of DP71. Flute. Sorry. I am ashamed because you are seeing all my results after, and then okay. I don't know. I don't know if they can make a special pointer, because I need really pointer for big, fat eyes. Okay. Here I summarize, okay? Even you see that even the expression of cadherin, that is the protein that is responsible of the interaction, they act as a molecule for the interaction between vessels and astrocytes. It's inhibited the expression, and that all the vessel density, the morphology affect even the delay of angiogenesis, okay? And now, okay, since, as you know, the interaction between Mueller cell, see, if you want, I think that you can put more light, okay? Thank you. We decided to see the blood retinal barrier, okay, was the permeability of the blood retinal barrier, since this interaction between the Mueller glial cells and astrocytes and the vessel was extremely modified. And indeed, that's what happens. We estimated the blood retinal barrier, and you can see that in absence of DP71, the DP70, the barrier became very much modified, okay? There is not anymore a blood retinal barrier. And we think that since the blood retinal, but the laminin, okay, the inner limiting membrane is the responsible of the permeability, then maybe the inner limiting membrane was also modified. That's what we observe. Here you can see in the wild type, okay, this is the laminin we without antibody against all the laminins, okay, and glutamine synthetase, okay. You can see glutamine. This protein is specific of myeloglial cells, okay. You can see that this is the normal inner limiting membrane, and this is in the absence of DP71. That means that certainly the permeability. This is the origin of the permeability of the barrier, of the blood retinal barrier. Here in with another methodology, okay, we observed the same. Okay, and now, since you know that, okay, when the blood retinal barrier in a lot of pathologies it's modified, it will affect, okay, the retinal vascular, and it will induce inflammation and capillary degeneration. That's exactly what we have with what happens in the DP71 null mice. Here in an scheme, you see all the results that we found here, the VGF expression, the ICAM, that is protein that is the responsible of the leukocyte adherence to the vessels, okay, and the degenerescence of capillaries. Here is shown that in absence of DP71, okay, there is increase of VGF. The receptor VGF doesn't change. It's just the myeloglial cells that produce more VGF. The ICAM, this protein, it's also it's responsible of the interaction of the leukocyte adherence in with the blood vessels. It's increased as well. This induces the vascular permeability and as well as the capillary loss. For that, with all these reasons, okay, you can realize that an absence of the P71, all this system is completely modified. These are all the, okay, all the phenotypes that we observe, but this is not the only phenotype. There is also one that interested much more you and of course Mirella, and because when she estimated the electroretinograms, the ERG, and I won't comment that. Maybe I will allow her to comment that, because of course I will say maybe not the right things, but you can see here that in absence of DP71, the response of the DP71 of the ERG, the big wave of the ERG was inhibited, okay, in the rods and on the cones, okay. That's also a very important result, okay. And if you have questions about this, you can ask me the other questions, but for this, it's Mirella that will come here, okay. And then, of course, with all this, now I think that you have, and we have also an idea. And all, I forgot just to tell you that since it's a history that I am telling you, that it takes some years, okay, during these years, and in the back of each results, because I took one results of papers, but all that has been published in several papers, in a lot of papers, okay, and they are the authors of the papers of the work in each. And I used to took a picture that represents what we were doing, okay. And also something that it's interesting is that the way in which, and I think that is research is like that. The way in which I present have a logic, but this logic was not the way that we did it. Okay, I take results here, and then now it makes a history that it should be continuous. As if we really ask each time okay, now we are doing this and we're going to do this. No, they become like that, and now, and that's very important for me, that I could build a history of the DP71 just in that is not a chronological one, okay, but now there's a logical one, okay, and I like to say so for you all the all the young people, the young researchers, because it's like that research develop, and I think that's for me, okay, when I realize that and I make the history, I think that it's the second or the third time that I present this story, and when I realized that, I was very, okay, I said, okay, come on, the research is like that, and I think that you think about that for you all the young people, okay? And now, just a last question that we ask ourselves: what happens with the DP71 and DP71, all the complex, okay, in retinal pathologies? And we choose two pathologies to show what happens with DP71. Pathologies that are dependent of the structure of the retina, okay? And we make an experiment, retinal detachment, okay? And we observe that if you see the result here, okay, this is in a control retina that I showed you that. This is Q4/1 and QP4, dystrophins, eutrophins, and beta-dystroglycan. And you can see that all the experiment, retinal detachment, that will modify the permeability of the barrier, of the BRB, the blood retinal barrier. It modified also the expression of Q4/1. Listen here, you see here Q4/1 here, and see what happens with Q4/1, what happens with AQP4. And what recall that is that the absence of DP71. And what happens when we estimated all the dystrophins, that all the dystrophin, the expression of dystrophins decreased a lot in the absence of, in the experiment with this experiment, okay? The eutrophins didn't modify. Why? Like it happens when the DP71 null mice, that means that the eutrophins are not modified, and that's what you observed here. And of course, the beta-dystroglycan that is here, okay? The beta-dystroglycan that I show you that in absence of DP71, it's not anymore localized in the neural cells like it showed here. And you can see that it's exactly the same that we observed. And when we estimated the DP71, we can see that the expression of DP71, that is this protein that, as I told you before, is the responsible of anchoring all these proteins. It's inhibited. Okay, this is the control. That's the DP71 protein in a Western blot, and you can see after the detachment, it's a really okay. Sorry. Yes, okay. I think I thought that I had. Okay, I put that. Okay, it's RNA and the protein at the expression are completely inhibited, showing that even that DP71 and all the complex has a function to protect the retina in a certain manner, okay? And then this when you affected the BRB by in another way by the retinal detachment, you inhibit all this the DP71 and all the complex. Okay, and we also observe exactly the same thing when we make a mechanical BRB breakdown. Okay, it's it's called the Irving gas syndrome. Okay, you can see that's certainly you know better than me that. But what we observe it was exactly the same. Did you see that after 72 hours, the protein, the DP71, the expression of DP71 was inhibited, but as well as the KIR4.1, the expression, it was not modified at all. But look at AQP4, okay? There is not anymore, almost not anymore AQP4 that. And that's confirmed what I used to tell you, that somehow this DP71 are the responsible of the functioning of the Müller-Glial cells, okay? And what was more interesting, but it will have taken too much time for that, but in this paper that has been done by Lourdes Siqueiros and Audrey, and another other researcher, the other girl, they showed that dexamethasone that inhibits the inflammation in retina, it's able to stop the DP71 is not anymore inhibited. It protects the dexamethasone. That means that all the system, we don't have the mechanism, the mechanism the yes, okay, but they show that the DP71 really plays a role in all these pathologies. Okay, and now in conclusion, because I won't show you all the things that I that's are the main things. Okay, in conclusion, the DP71 deletion in mice alters the localization and expression of KIR4.1, AQP4 channels and ducts in the membrane of Müller cells. It alters the macroglia and the vascular morphology, induces retinal vascular inflammation, capillary degeneration, and alter the integrity of the BRB. And they perturb the ERG responses. I think, okay, and let me tell you that for that we have a very nice paper, and we will maybe discuss after. And moreover, with Mirella, moreover, moreover, the experimental retinal detachment and the mechanical BRB breakdown are associated with the DP71 downregulation. Okay? Okay, and with that, I show you all this previous story. And finally, at a certain moment, we decided to tackle the next, okay? What was next at this point, okay? To produce an AAV, another adeno-associated virus that will target specifically Müller cells and to try to restore DP71. That was something very difficult that Ophélie Vaca, that some of you know, okay, she was fighting with that because, you know, okay, to find a virus that specifically tackled Müller-Glial cells was very difficult. But we were very lucky because a research, a postdoc in the United States came to work to our lab at the Institut de la Vision, and this girl is Denise Alcaraz. Maybe some of you know about her. Now we're still collaborating, Cyril and ourselves, we are still collaborating with her. And she came with a virus to work in our institute to work with a virus that specifically target the Müller cells. Of course, that happens, and that they have enough place inside to have a protein of 71 kilodaltons, because the protein is still there when we started that, were smaller. And then that was really in collaboration also with a Mexican group. We make really this kind of network of work, because with the Mexicans we succeed. They are very good molecular biologists. We succeed to synthesize. Okay, I show you the experimental protocol that we did. Okay, then we synthesize a green fluorescent proteins and DP71 together, and we succeed to clone it. Okay, and then introduce to an AAV SSH12 and produce this AAV and make an intravitreal injection in vivo and make the observation during two months with the fundus of the eye and OCT, and we analyze the effects in the expression level of DP71 and DAPs, in the localization of these proteins of DP71 and DAPs, and also in the BRB permeability. And what were the results? Okay, here you can see this is the first result that I follow. We follow here the expression of this complex SSH10 with the GFP at DP71, and you can see that is the fundus of the eye. Okay, that is already green. That means that when you make this cut here, you see that it's localized. The EFP, that is it has been localized in the mural glial cells. This is the mural glial cells, and this is a merged image. That means that this complex that include inside a green fluorescent protein, the DP71, really from the mural cells, it penetrated and it was localized in the mural glial cells. Okay, and what happens with the DP71? We were also very lucky. Okay, you can see here the expression of the mRNA of the DP71 in the wild type, in the absence of in the DP71 mice, and after the virus transfection, and you see that there is an overexpression of DP71. We don't know yet why. Okay, that's till now neither of the molecular biologists to which we are working okay, have an answer for that. I am not doing any more experiments in my lab, and I don't know if once I will have the answer, but not the mRNA was highly overexpressed. You can see, okay, the difference. This is normal, and this is after, and this is the protein, okay. The DP71 neural, there is not, and look at the amount of protein, okay. And in spite of the fact that we have all these proteins, what happens with the rest of the DAPs? Okay, you can see here that we also found an overexpression of the mRNA associated with EP71. You can see here what I showed you before, the expression. Beta-dystroglycan was the same if you compare, and eutrophin as well as eutrophin was overexpressed, AQP4 and T4.1, the green fluorescent protein as well, laminins as well, and agri. And it seems that there is a complex that DP71 maybe it restores, even if we have a lot much more of DP71, and it was not other point is it was not a DP71 modified. It was not an isoform of DP71. It's the full DP71 that we introduced, and it was not okay. And that's something that okay, what we succeed to restoring, but the exact molecular mechanism that are under this observation, this important observation, is not known. I don't know. Here you can see, okay, what I am telling you with some examples. Okay, you can see that here, of course, the wild type DP71 in mice and DP71, okay, in the with the expression of the DP71, you can see that is located exactly around blood vessels in the inner limiting membrane, as well as the beta-dystroglycan. Okay, it is after the DP71 restoration. It's located as well as AQP4 and Q441. And you see there is AQP4 much more AQP4 than the normal mice, okay, and Q441. And in spite of that, everything is functioning. And it is not only the expression, the function of the DP71 was completely restored. You can see, okay, if you remember BGF, the expression of BGF was highly increased in the DP71 null mice, okay, and in presence of DP71, completely we came to the normal level of BGF and the blood retinal barrier. Something, sorry. Okay, something so complex as the formation of the BRB, of the blood retinal barrier, was completely restored. Okay, that means that in these 2 months after the transfection of DP71, we succeed to recover a functional retina. And that's something that, okay, for me, I still be surprised when I expose that, the results. I say, okay, we were very lucky with that, okay? But not only that, now, Mirella, we've published a nice paper with that, okay? You can see that also the ERG, okay? Such a complex, okay, that has been modified, it has been recovered completely and after the DP71 expression again in 2 months, okay? I hope that maybe somebody injected me a little bit of DP71, I will become younger, okay? Okay, then now with this, okay, I see that the end my talk, maybe, okay, I hope that you still, okay, reading, but because in conclusion, our data show that the SSA10 GFP DP71 induce intense absorption of DP71 in the DP71 neural retina, adapts overexpression and relocalization of the dystrophin-associated proteins, a BRB restoration, and an ERG restoration. That was very important. And now you will allow me to read something that I take a little bit inspiration of what, because with all these results, part of these results has been summarized, okay, and show in a very nice review that we write. And then this is the message that comes from what we did. I will read it, okay, because I cannot say that, or you can read it, okay? To our knowledge, we report here for the first time the neurophysiological deficit of the retina, considered a classic non-muscular symptom of DMD, can be successively reversed by gene therapeutic intervention using NAV vector subtype designed to specifically target molecular cells. This encouraged the use of DRG as a reliable, non-invasive method for the rapid determination of gene therapeutic efficacy in ophthalmic conditions. ERGs may also be predictive for the success of therapeutic interventions in other parts of the CNS, and future experimental approaches that test the efficacy of DMD treatments in the CNS may use retinal physiology as assessed by ERG as a valuable biomarker. And also, and now I will really thank my colleagues and all the colleagues from Brazil, the colleagues from Germany, and the colleagues of France, okay, that these results, even if I had already 10 years of retired, has been published in one of the most important reviews in the field of ophthalmology, okay? The Progress in Retinal and Eye Research. And you can see that the list of I think that, okay, that was for me, it was really, I feel, how can I say? I said, okay, Alvaro, what you did with all your teams and with your collaboration, it really okay, it's in very good hands. He has been included in this important. It was not a review of the P71, but the part that we did in the mice was included that, and I really thank you very much indeed here. Okay, and I think that now, okay, this is maybe a small list of the collaboration. You have been all the collaboration that in which we develop all this work in the Weizmann Institute and with Andreas Reichenbach from Leipzig in Germany, Cecilia Montañez in Mexico, Dominique Mournet in Montpellier, Michel Pack in Paris, Schaefer and in the University in California, Berkeley, and of course Dora here in Brazil. And I acknowledge, okay, I have all this list of acknowledgements, and maybe it can be even more, okay? But I thank you, all my collaborators. And to now, I will ask you something, okay? I will just make a parenthesis, okay? You know, there is somebody that is here, Ramita Yoni. He was one, and I call him in past. He was one of my good, very excellent student of PhD. He has an MD. He became a PhD. He became after the PhD chief of several clinics of ophthalmology in two big hospitals in France, in Paris, in the Rothschild Institute, in the and in two other hospitals, and they make the French government make create the Institute of Myopy for him, okay, and that was it was last year, and unfortunately here in this now in Sao Paulo, he died of a heart attack in the month of May here, because he became here to Sao Paulo because they have a distinction to give him, and they found him in the hotel, and I really I am telling you, and I really feel very the emotion because it was a really bright student, like people like I stopped there, but I wanted to have a thing on him, because I am in the town in which he died, and he was also he was expected to succeed José Alain Sahel at the direction of the Institut de la Vision, and of course life at 50 years old was taking anyhow. Now to end my talk, I will talk about 10 years ago. I cannot that. You can see. I'm sorry. Maybe if you can just to see me better the picture. I found these pictures and I was so happy. To find it. You can see there. I think that Mirella is just at my left. Yes. Yeah, sure. That's well. I had and of course, I wanted to make an honor. I think that I would say an honor. Le voy a honorar la presencia de Dora Fix Ventura. And now the retina has a window of the brain from my research. To see the disorders, you have to wait to Aurélie and Cyril to talk about that. It Wednesday and Thursday. And muito obrigado pela sua atenção. Atenção. You have questions? I still haven't. I don't know if I can we put slide again? Let's see what. And of course, I just want to say that I make just I arrived 3 days ago, and I have before my old friends, I have new friends. I have in the order of Joe, okay, Sarah, Andre, Julian, and Sergio. Okay. Sure. Okay. Thank you very much for inviting me.
Thank you for coming. Yeah.
Oh, thank you very much. You know, I hope that you will arrive at 80 with the energy of Dora and maybe mine also, because I will be 80 in 3 months.
Try 8.5.
Are there questions, or we can just talk after questions? We have a question. You need to come here. Sorry. Wow, he has a big list.
Yeah, I actually have 3 of them.
Otherwise, I will be stopped. I will be sick.
Professor, thank you for your lecture. It was really amazing. And well, as you know, I'm a kind of beginner in this world of electroretinogram as
But
well,
you
and
have, also something of Mexican.
so therefore I have so many questions. The very first of all is, do you have any hypothesis about the relationship between the size of the DPs, of the dystrophins, the different dystrophins, and their function, especially in the retina?
Of the size, no. I don't think, no. Really, from the sides, of course, the biggest dystrophins, as Cyril know, and then you know, and with Michel, who we are studying all the, and with you now, okay, the expression in the synapses between the photoreceptors and, but I don't know how. Just they went into the mural cells and the astrocytes due to the size. I don't think so. I think that it's an evolution. It's a question of on evolution. That's I don't have. I don't know if you can help me, because yes, I don't have an answer for that question.
Maybe not really a complete answer, but maybe one distinction is the full-length dystrophin has a lot of repeated domains that gives the protein some elasticity, which means that this dystrophin is not only a structural protein to rigidify the membrane, but on the contrary, to give some flexibility during a contraction of the muscle. So that could be at least one function that is not needed maybe in glial cells for clustering aquaporin receptors. But then this does not explain why there are several sizes and what are, but at least for the full length, we could say that. Then you may
And even,
even for the 260.
it's already
Yeah.
big, it's very big.
And then some of these proteins, they lose some functional domains. There's not only actin binding domains in the different region of the dystrophin, but there are other functional domains, interaction with cholesterol, interaction with many different types of system and pathways, and maybe some of them are needed in some cells, not in other cells. That could be another reason for having or not the full length of the dystrophin. You may say, oh yeah, but why the full length is in neurons? There's no contraction in neurons. However, it's known that when you have changes in the extracellular homeostasis, and they may have some change, some Yeah, kind of a contraction of the membrane of neurons as well. And it's been shown that's a complete different literature that contraction of a neuronal membrane can modify the function of some very important receptors on channels like NMDA and AMPA receptors. So possibly, perhaps, but this has never been studied, perhaps there is a link between the need to have the full-length dystrophin and the need maybe to have some elasticity in the membrane movements in some specific events. But that's not a complete answer, but maybe some ideas that have not been very much studied so far.
Thank you.
Maybe yes, but something that I forgot to tell you, because it's quite complicated, and then maybe that we can ask to Mirella or to Jan to answer the questions. How for me, I didn't understand, since I am not an ERG man. Okay, how come you can restore with DP71 the ERG? And I didn't understood well the influence between, for me, at the level of the plexiform layer. What doing there? But he proposed, Jan, he proposed a mechanism very interesting, that is the control of the potassium and water, and for explain how, in the absence of DDP71, but all the other proteins are still working, okay, there in the outer plexiformal layer, you have a big wave that are inhibited, okay? And of course, we thought a lot with the observation in the DP71 null mice could be related to the cognitive night problem in the eye, okay, and that's not true. Okay, it's completely different.
Well, my second question is also related about this electrophysiological relationship between the dystrophins and the different cells in the retina. A time ago, I was discussing with Mirella about the possibility of being a relation between the lack of some dystrophins and the regulation of the calcium waves, or even, for example, in the case of the synapses between the photoreceptors and the bipolar cells, in the case of the calcium channels. Do you think it will be a relation? And even more, because you say that the different dystrophins, the different lengths of the dystrophins could be related to this plasticity, maybe also be related to the conductance and the capacitance, the difference of the capacitance of the membrane at this point.
Yeah, maybe it could be for the same reason that discussed before, but I think there's not much data on calcium waves and channels in retina. Maybe Alvaro, you know, but at least there are some papers showing relationship with calcium homeostasis in the muscle. There are a lot of papers on that, and in the nervous system, I don't know it. In the retina, it should be studied, maybe. But for example, there's a recent paper suggesting that at least in the brain, DP140, we don't know much about DP140 function. It seems that it could be expressed at the presynaptic site in relation with calcium channels. Bon, but so far, there's not, I think, no more data on that. Aurélie, no. Yeah, so calcium has been mostly studied in the muscle, and there are old papers from the 90s showing calcium alteration in the cerebellum of mice lacking the full-length dystrophin. And so muscle, brain, retina, maybe it's...
The Mexican Dalila Martinez, she showed that there are some relations on the on calcium entrance to the mitochondria and the DP71, because in brain, DP71 is located in the mitochondria. So something that's okay, but it was just a first paper, and it was not even a paper. It was a paragraph in a paper that just really showed that it could be a function, but more no. I think that's it's a good field to develop. Okay, I think absolutely.
So there are some clinical trials with Benoni, which is a medication for Leber hereditary optic neuropathy. Yes, to treat mitochondrial dysfunction, and they are trying with Duchenne patients as well because of this. Mitochondrial DNA. Mhm.
Thank you, Professor.
Okay. Come on, give your question. Don't be shy. I was not shy to talk with you. Yesterday we have a good evening. Yes?
Thank you, Álvaro. Hello. Hi, I have a question about, I don't know if you understand very well. I think in the study of Barboni, how long after gene therapy you perform the ERG? You have it was made a longitudinal study? Yes, it's a good one. 3 months is a good one. So thank you. It's only this.
Good question. Good question. Thank you.
Thank you.
Do you have experience with the expression of CP71 in other primary retinal diseases? We showed a paper about the reduced expression of CP71 in retinal projection. Have you looked into other inner retinal diseases?
Very good question. We were dreaming about that. Okay? And Cyril and myself, we asked for a grant with a friend of us. I won't tell his name. With the diabetic retinopathy. With two pathologies. With the one that is the I don't know how to call that, the cecity. Okay, problem of cecity, but the main was the diabetic retinopathy. In spite of that we get the money has not spent by this our colleague to answer this question. Patrice Ford did. But Patrice did his PhD with me as well. Okay, come on, the word is little bit, okay? Okay, please tell him, and then we send him, because I saw him in the Arvo last year, and then, okay, this year, okay? That's true, but that's nice, okay? It's small, but he was not the one, okay? It was somebody of the Institut de la Vision that got the money and didn't make anything. Okay, that's the problem. But I think that for that, it should be links of that. When you see the mechanism, the molecular mechanisms that induce the diabetic retinopathy, I think that the DP71 should play a role, for sure. Thank you for your question.
No more questions? Oh, please come again.
In fact, we discussed so many times together, Alvaro, that's sorry, but it's more a question for the talks of Dora and Marcello. So, because that's a question session for all the talks that we had today. So, well, Marcello is not here to answer, but I was very impressed by all the battery of tests that he developed to address cognitive function or neuropsychiatric comorbidities, which are also present in Duchenne muscular dystrophy, such as attention deficit disorders or autism spectrum disorder. So I was wondering two things: whether this has been already tested in DMD patients here in Sao Paulo, and the other question is, how do you think this kind of dysfunction could this affect the optomotor reflex? Which is also involving, I guess, a lot of brain processes for integration of vision. I don't know if you have an answer on that. But.
Well, did I understand correctly that you're asking if the new tests have been used for DMD patients. Well, not much. Not much, because we have been concentrating on the new electrophysiological tests, but the psychophysical tests have not. So we have indeed used Mirella's in Mirella's doctoral thesis, the tests developed by Jan. Actually, she went to Jan's lab. That's a story. Can I tell the story? Yeah, well, I'll tell you in private. And but it was a very successful stay, very up to today, and so that is the part we have developed. But so far, we have not used any of Marcelo's tests. It would be very interesting to do that, especially in DP71 patients, which have a greater effect in the nervous system than the other patients. Actually, I was going to ask you about the rescue of brain DP71.
Thursday.
I will talk about that on Thursday a little. Yeah. But another question, in other diseases where brain dysfunctions are present, do you know if there's any impact on optomotor response, even if vision is not affected, for example?
Well, we are looking at the optomotor tests now, the behavioral tests, and I don't have the results yet. So, but we will very soon, and we expect to do these tests in zebrafish also. Yes. Okay, so that's a good thing to do, but okay. Now you have the floor.
A last very small question. I see that several people here, Mirella in Budapest and you here in Brazil, you have a good experience now with the retrieval portable system for ERG in children. So finally, do you use skin electrodes or other type of electrode? Because in my memory with the skin electrodes, the responses were much smaller, not very good. And I think maybe we will soon start to use also the retrieval in France for DMD patients. So I think that's a good point of discussion, maybe to know where you are with your experience with this.
Yeah. So yes, in now I gave up using the skin electrode, but at the same time in Budapest we are comparing, making a large group for comparison. Just from observation, I think the problem is always the off response. For some reason, it doesn't come with the skin electrode. I don't know why. But I can understand that if you want to test small children, it's a bit hard with the DTL. But here we can, in children 7 years old, tell me if I'm right, Alodia, what was the youngest you could put the 9? Yeah, 7, 8 is a little bit hard, but 9, 10, it's already fine because actually doesn't hurt. It's just a bit more invasive than just the skin electrode, but the DTL, yeah, DTL electrode.
Big problems, behavioral problems. Can they stay long? I mean, can they? Do they behave well? Do they behave well when they have to be dark adapted for half an hour and then tested for? I mean, a session should last 45 minutes, maybe, or 1 hour almost. So, or is there some genotypes? That's a discussion. Yeah, I mean, is it easy to do the whole session, even with patients maybe lacking all dystrophin and being autistic, or?
it's very difficult to do that because all the protocol expand 3 hours or 4 hours, not only ERG, but in the beginning, and we talk, we explain, and then we do some, we evaluate the visual acuity, and then dark adaptation, and then pupillometry, cognitive tests, QI, and the ERG is the last one. So some of the patients don't want to do that, or because the electrode and they are scared about, or just about they are tired, and then, but ERG spends about 1 hour since the first preparation until they finished, and the dark adapted is 20 minutes, and we do yeah, and it's a very long protocol, and we do the light adapted and dark adapted in these patients, and it's very long protocol.
So yes, the ERG has to be the last. That's the problem. If you are just doing ERGs, I think it's another situation, because then you start with your patient doing the ERGs. But here in our work, we need to leave it to the end because you dilate the pupils. And I think, yes, the dark adaptation is a bit hard because sometimes the kid doesn't want to stay 20 minutes dark adapting. But if you start to tell stories, talk, and ask what is your football team and what do you like to do, and then usually it goes faster and they can do it. But what we saw is that the ones with the alteration of all proteins, they are the most challenges, because we had about 10, I guess, but the ERGs were done with 3. I don't know. I don't remember the DP71 patients. Three, yes. But I think we had here like 10, more or less. But they are, yes, they are the ones, yes, a bit more than the others, yes. Not necessarily, yeah. Maybe Sarah can say something you would like to say. Sarah, Leo is asking if the DP71 patients, they are more autistic than the others.
Don't cry, don't cry. Go ahead, Sarah.
Thank you.
One, two, three.
The patients with DP71 have more problem with the cognitive problem and autism and the TDAH, ADHD for 20%. 40% has a autism and TDAH and cognitive problem. In my test, I have 14 patients with alteration in the P71, and 7 patients has a problem with cognitive problem. So in my student, 50 have problem. Yeah, severe intellectual.
Let me ask you about brain DP71 rescue. How would you go about doing that?
In the brain, I didn't. It was Cyril that did it.
Yeah, but
Okay.
how did you do it? Did you inject in a specific place, or did you have it circulating?
Yeah. But for it depends. We did several papers with different dystrophins, but for DP71 we did this recently, and in fact we used two different ways to deliver the vector. Was with a vector. It was not a vector specific for Müller glial cell. Was a different serotype, and we injected either in young adult mice in the ventricles, so it can circulate with LCR. Or we also did another experiment where we injected in the heart, but in newborn mice, and because at that time the brain barrier is not completely formed, and it was very early, P1, P4, and so 4 days after birth we injected so that it could also distribute well in the brain, because the brain barrier is not fully mature at this time. But the best results were rather obtained by injecting in the ventricles. Yeah. No, we did not try in very old animals. We tried in mice. I don't remember exactly the age. I think they were late adolescent or young adults, which is more or less translational with the idea that, I mean, if there's, we looked at behavior changes, we looked at the molecules that, the channels that Alvaro talked about, because they are also clustered by DP71 in the brain, not only in retina, aquaporin 4. Yeah, but in fact, we just looked at aquaporin 4 on syntrophin. No, no, in one experiment it was not an overexpression. In another one there was an overexpression. So it depends on the type of delivery that you use. But in any case, we had the problem that aquaporin was restored partially, but behavior was apparently not improved. So, well, we have several questions around that. Maybe because it's too late, or maybe because we inject only the full length DP71. And as Alvaro was mentioning, maybe at some specific moment in the brain development or plasticity, we need to inject specific isoforms. But as Alvaro showed, there are like 12 isoforms of DP71. Which one is the one needed to have a therapeutic effect? Yeah.
Also, concerning your okay, concerning your talk, I think that we were very lucky in the retina. Why? Because as I show you, the inner limiting membrane that allows the passage of the virus. Okay, we don't have to. We make an intravitreal injection, and not the other one that is most destructive for the eye. Okay, and then with that the virus passed very easily. Okay, and that's what we were very lucky, because we didn't expect to that. Okay, and of course we even have a patent that to try to allow all the virus to penetrate, is that with an iRNA you inhibit momentarily for a moment the expression of DP71, the membrane, the inner limiting membrane becomes permeable, the virus can go in, and then after it close. But okay, I didn't have a succeed with the patent, but that was our really lucky fact. The fact that virus go like in their street, okay, they just go directly because the linear limiting membrane is permeable. It's not the case in the brain. Yeah, it is not the case in the brain. Maybe that.
The barrier is also altered in the brain, but maybe not in the same extent or way. Yes.
Yeah.
Okay. So I think I will ask if any more questions.
Oh, one more question.
In dark adaptation, the patient's DP71 is the threshold is simulans control, and other DPs is different. We think about the compensatory mechanism. What do you think about?
She made me say the question in Paris. But that's, yes, it's very hard. It's very hard to say, because how can we compensate to that? Because that's so astonishing. That's what. That is the reason when Cyril told me there is a girl called Sarah that is coming to the show the dark adaptation, and she showed that the DP71 patients they don't show any problem, okay, and the other ones yes. How? Okay, I think that I really don't have an answer. Okay, at least maybe Cyril has one. Okay, but no. Okay, and what is not compensated? Do you know, Cyril was my graduate student 30 years ago, and he's giving the answers of me. Okay, thank you, Cyril.
No, it's not really an answer, but just one thing is that this kind of bizarre result, we have it for the behavior as well. We have a model lacking all dystrophin, showing less problem than those that are lacking other dystrophins, but still having DP71. Okay, so the problem seems to be not only for retina in the patients, but it happened also in one of our models, in which we have looked at one possible compensation that we could think is eutrophin, for example. So we looked at eutrophin in the brain of these mice. It's completely normal. So it's not compensated by eutrophin. That's all I can say. It's not eutrophin.
Yeah, that's okay, because we observe the same. Okay, the eutrophin, it doesn't move after when you inhibited DP71, and there is not a compensation. Eutrophin in the case of retina cannot compensate for the absence of DP71. That's for sure. No answer. No answer. It's very difficult. I was so surprised that I took the next metro and then I went to listen to Sarah talk, because I was very surprised, because for me that was okay completely. With other things, absolutely, completely. Well, but it goes into the idea that DP71, although has a function in the brain, it has a completely different than in the retina. Okay, and the retina is located just in two specific cells, and when it's not there, everything gets broken. Okay, but not in brain. In brain is more, but the behavior is modified in the cow DP71, in the men that don't have DP71, and is modified. That, as you know, in that case there is a contradiction of that. Maybe that is the ways that there are nerve ways that are different in the men, and I don't know. I really don't know. But can you do the same experiments in the cow DP71? That's what I propose you. Do the same experiments with the cow DP71, and we will see. Haga los mismos experimentos en las cow DP71. Okay.
More questions? Jan, come, please.
Okay. We were missing.
I mean, it just appeared when I heard your talk about evolution. If I would construct something, or I would put it in a different way, I would think that there is a large evolutionary pressure to keep the X chromosome as small as possible. Why isn't it? Because, I mean, if it's on the X chromosome, males are always, if there's something wrong, then it's expressed in males. And with diseases like Duchenne muscular dystrophy, it's quite how should I say, it's a catastrophe, and they are not able to reproduce themselves. So why not putting it on another chromosome? Why keeping it on the X chromosome? And it's not only with DMD, but it's also with congenital stationary night, retinoschisis.
Because it's also okay. Because also the congenital
stationary night blindness.
That's also in the X chromosome. Sometimes we think that it will be with Christina sites and almost.
And the retina can be quite dystrophic when there's CSB.
Yeah.
That was just a question.
Yeah.
No answer.
Yeah, I was thinking about the negative ERGs in the patients with DP 260 altered. So you know if there is some study about the gene therapy with other dystrophins, other DPs?
The retina?
Yeah.
No.
No.
There are not. There are not.
Thank you. Another dystrophins.
We are just starting to do it. This is our ongoing project with Aurélie. We try to rescue the full length and DP260 in mice that are lacking those dystrophins, but for the moment it's just that's the project we do now. Yeah.
I think that now there are viruses that are bigger, and then you can put these two DP260. But already when we did that with Ophélie, that okay, just for you, Ophélie was a student of mine, then he was a postdoctoral with Cyril, and now she's working with Aurélie, okay, and that's the same, okay, girl, and then maybe she will succeed to put DP260 in a bigger virus, and then they will restore. That will be nice, okay, but at our time, to do that with DP71, that is the smaller one, it took a big effort to do it. Okay, now it's much more easy. Okay, and then maybe they will succeed, and we will see that the cow DP260 will recover it. Okay, who knows? At the brain or at the retina? What happens with them? Oh yes, AOG. After yes, sure. About the data that I show, no, we stop it there, okay? And okay, because for several things, you know, as I told you, emeritus in France doesn't have the right to have students, no money, no leader team. And Ophélie was one of the last students. I just stop it. And dear G's. No. He was not. Otherwise, we could do it with him. But this was long time ago. But I think that maybe they can do it. Yeah, maybe. But I don't have an answer. Unfortunately, it has to be done. There are so many things that should be done. Okay.
The DP71 patients, when have a cognitive, it's okay. Then compare it with other patients, is the good behavior. So sometimes study and work and other patients is limited.
Yeah. Okay, I didn't catch very well your questions. you said that in the cow DP, in the main cow DP71, okay, it could be a better delivery of the AAV, of the virus. Maybe, I don't know if we will never try to recover it with the AAV, with our AAV SSI 12 with in men, okay?
No, but there is no indication because they have normal visual acuity. So you wouldn't do a treatment specifically for the eye in Duchenne patients because there is no visual, like evident visual loss. Yeah, you don't give an injection, for example. So what can happen in the future if the treatments from Aurélie will, of course, succeed and go very well to the central nervous system is that in a systemic treatment for the patients, you will have the retina rescue as well. We hope. One question also, because behavior. And then you need to ask to Cyril again. Yes, wait for Thursday. He will explain on Thursday.
Okay. Maybe the way of doing this meeting, it will be Alvaro, Cyril, Aurélie. Okay.
Yeah. Is it for sure in genetics that if you have the alteration before the exon 63, you will be with your DP71 normal and after affected? Like, is it expected that in patients this is very straightforward? Or would you expect that maybe the patient has multiple mutations? Yeah. It's rare. Okay. Because I think the DP71 patients, they are kind of different from each other also, at least in terms of ERGs. I know the dark adaptation looks better for them, but in ERGs, you can have the ones very similar to the DP260 patients and the ones with relatively good ERGs. So I don't know. We'll see.
You mentioned that there are different isoforms.
There are different isoforms. I will never test it. They have to be tested, all the isoforms.
Yeah, but that was why I asked about the genetics. So once they have the mutation after 63, they will lose all the isoforms or not for sure? I don't know.
Sure, of course. All
yes, all.
yes,
Okay.
all of them.
Yes.
Yes, that's for sure.
Yes. No, let's go.
Dystrophin disappear.
We need to go. That's the message. Okay. Thank you so much. Que bom, porque eu não sei fazer isso.
Ah, okay. 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 Thank you. Yeah. Because I don't know. Okay, okay. I will say what
Uns bonecos novos, hein? Umas coisas novas, modernas.
Mas ainda não tá. Eu
100%.
eu continuei nos eventos até agora, quase 7 anos. A gente tá pedindo algumas coisas.
Mas você tá fazendo o quê? Vocês estão fazendo isso daí virtualmente ou vocês têm que
Não,
vir lá?
tem dois. Esse aqui é o micro de transmissão, que eu vou desligar lá na sala. Esse aqui é o de apresentação com 2 câmeras.
Não, já morreu. Isso aí já matou, né? Já desligou.
Já.
A câmera tá ligada lá.
Tá.
Desliga?
Eu vou desligar ela, vou lá no fundo, desligo ela manualmente e desligo a coisa.
Pode ir. Pode ir. Que eu não...
Você veio. Você veio trazer o professor?
Não, eu vim pra ver.
Ah, que chique.
Convidado. Ele tava lá em casa.
Hã?
Ele tava lá em casa.
O professor?
É.
Ah, você conhece ele?
Sim.
Ah, que chique.
O Álvaro e o Cirilo. Os dois estavam lá em casa.
Ah.
Vem
Não,
pra
mas
ver.
você tá ligado no.
Não, mas não.
Ah, tá. Você tá ligado ao behaviorismo?
Como é que é?
Behaviorismo, sei lá.
Bevarrolismo. Não, mas eu não falo francês.
Hã?
Eu não falo francês, nada. Né?
É, agora que eu olhei, porque eles pediram esse evento quase em cima da hora. E aí.
Nossa, meu, já tem uns 15 dias que tá programado isso.
Sim. E...
Eu até achei que fosse lá que era mais fácil.
Não, hoje teve congregação lá. Teve congregação, ele não deu.
Eu não gosto de evento aqui nessa sala. Nunca gostei.
Terminou quase agora. A gente pegou a mesa da. Isso aqui era tudo da pós, trouxemos pra cá. Trouxe pra cá.
Da pós?
Essa mesa foi feita.
Essa mesa foi, faz tempo.
Faz tempo, aí a gente trouxe pra cá, mas o resto continua mesmo.
Continua mesmo.
Só algumas coisas que a gente mudou.
Fizeram cortina agora.
Ah, é. Eu pedi essas cortinas aí. Veio. Não tem jeito.
Essa bosta.
Não, e o pior é quando elas caem.
E esse banheiro de shopping?
Você achou bonito o banheiro? Foi.
Banheiro de
Foi.
shopping.
Deixa eu ver. Como assim, banheiro de shopping? Você achou bonito?
Aqui, o banheiro daqueles prédios.
Ah, eu entro um pouco aí.
Você nunca foi no banheiro ali?
Não, já fui. Mas eu entro muito pouco.
Eu
aí.
também, mas eu entrei a primeira vez. Ué, fizeram um banheiro de shopping aqui pra ninguém. Não tem aluno.
Tá pior. Aqui não tá. Os professores pararam de vir e os alunos. As aulas estão quase todas online. Um curso integral e as aulas virtuais. Ah, não tem mais aula de pós, né? Todas virtuais. Não tem mais aula de pós. Nem aula, nem reunião.
Acho que nem de
O que
graduação,
só.
porque a hora que eu cheguei aqui,
E as de graduação são muito poucas que tem.
eu não vi ninguém no prédio. Ninguém, só vigia.
Só o
E
vigia.
outra, quem foi o idiota que mandou pôr uma porta corta-fogo de vidro?
Eu não sei.
Yeah. We have to work with the close.
Então, vai lá, se liga remoto. Pode vir, mas a gente quer continuar, tá muito gelado. Quinta-feira eu vou chegar mais cedo, aí a gente se fala, tá? Vai ser aonde?
Amanhã vai ser aqui. E eu acho que vai ser na sala de 18.
Na quinta?
É, não é todos os dias aqui.
Quem colocou essa bandeira aqui? Tá errado.
Não sei, tá errado.
Porque essa aqui de São Paulo é desse lado e essa aqui é desse lado. Depois eu digo. Ó. Não é seu não, né?
Não. É de alguém daqui.
As coisas estão tudo aí guardadinho. Tá. Bom, eu vou voltar na quinta-feira para assistir a palestra do Cirilo. E aí eu vou ter mais tempo para a gente bater papo.
Beleza. Mas depois que ainda tá do mesmo jeito. Por exemplo, essa